Okay guys, so let's talk a little bit about these operons. This was one major difference between eukaryotes and prokaryotes, and that is that prokaryotes, bacteria, have these operons, these sequences of coding regions, genes basically, that all have to do with the same basic function, and they're all controlled in one fell swoop. Okay, they're all produced at once. Okay, so we could be talking. Cell products come in many different forms. All right, so if we think about enzymes that are produced in a cell, let's say these enzymes are needed all the time. Okay, we might see that they are produced all the time. The genes for the production of the RNA and then ultimately the proteins would always be producing a certain number of those proteins, right? So they're always being read. What we'd say about this is that this is a constitutive production of that particular product or products, okay? It could be an enzyme, it could be some other product, okay? But they're needed all the time, they break down, the products break down, and you need to continue to produce them, so constitutive. Now, there are some products where you would never want to make those products unless there's a certain condition that is met in the environment. Okay, so to give you an example, if I'm a bacterium and I have the genes necessary to produce enzymes to utilize a certain sugar, but there's none of that sugar around, why spend the energy and the time to produce those products, right? I want to be able to induce the production of those products only when the sugar is present. Inducible genes or inducible operons can be induced and turned on to produce products, okay? Now, repressible operons, sequences of genes now that are repressible, this would be for the production of a product, let's say, that's very energy consuming. You don't want to, you only want to make that product again when it is necessary, okay? But in this particular case, it's going to be turned off in an active way, right? So the difference between inducible and repressible would be, inducible would have to be in some way turned on by the presence of something like a sugar, and repressible is got to be turned off when there's enough of a certain product produced. Okay, now this may be a little confusing right now. I think constitutive is fairly intuitive, right? Constitutive means it's constantly made, it's needed all the time. Inducible and repressible, we don't want to have these genes on all the time, and what I'm going to do with you is go through those two in detail, all right? Now, to show you an example that can help to make the point here, these two particular plates are from an organism called Serratia marcescens, okay? Serratia marcescens is on both of these plates, both TSA plates, no difference in the plates at all. What the difference is here is there's a difference in where the plates were incubated. In the case of this nice pink kind of beautiful color over here on the left, that was incubated at 24 degrees, right around room temperature. Versus over here, where we have no color at all, it's pale, right? It's off-white. It doesn't have that color. So this is just one example where a variable in the environment, in this case temperature, was able to turn on a set of genes that made a pink-red pigment in a certain condition, and it wasn't made in the other condition. So this is just kind of the idea. We're going to be able to turn on and off these operons, okay? So let's start our first example. The regulation of transcription is going to be with a Lac Operon first, okay? So Lac Operon refers to lactose, okay? Let's just go over the basic parts of an operon, okay? We have a promoter. P is for promoter. This is where an RNA polymerase will bind first to get ready to transcribe, right? So the initiation process for transcription is the RNA polymerase binding. That's where it happens, the promoter. Now, oftentimes, right after the promoter, we find a segment of DNA called the operator. Okay? So here we have an operator, segment right after the promoter, and then I see 3 genes, Z, Y, and A. These are genes for the use of lactose, the sugar lactose. Upstream from this sequence, right? Everything would go in this direction if this were to be transcribed, okay? Upstream from that, we have something that is called a repressor gene, okay? The repressor gene is always transcribed. So here we have transcription of the repressor gene. We get a transcript, a messenger RNA, and from that we would have a translation product. So here it is, okay? Now, in this particular condition, in this particular slide, there is no lactose present, okay? So, what happens with this product of the repressor gene? Well, this particular product, when there is no lactose present, it's a perfect shape to fit right up here on the operator. Now, this is pretty much completely in the way, right? If an RNA polymerase binds to the promoter and it wants to ride the train tracks and go down this and read to make a transcript, it will not be able to do so because the repressor protein is sitting in the way. It's sitting right on that operator, inhibiting that from happening. So, when there's no lactose around, there are no genes read for the use of lactose, okay? This is an active repressor protein in this scenario, right? All right, now let's look at the lactose operon again. Okay, so again, this is our Lac Operon. Nothing has changed. We still have our Z, Y, and A genes. We have a promoter, we have our operator, we have our repressor gene. Just like before, the repressor gene produces an mRNA for the repressor protein, and a repressor protein is produced. In this case, however, on this slide, there is a lactose molecule. Allolactose, don't let it mess you up. It's just like having a lactose molecule. So, in this case, lactose is present. Lactose is in the broth tube. Lactose is there with the bacterium, okay? And the lactose would act as an inducer, okay? So in this particular case, this lactose right here is going to bind, and it's going to take up this space right here, okay? And this would change the repressor just enough so that it is unable to bind at the operator, okay? This here would not allow the repressor protein to bind. Our RNA polymerase was able to bind, so it started back here on our promoter, right? And there's nothing there to stop it. So it went right over that operator and transcribed those genes, and ultimately they were translated, and we now have the products necessary. These are all having something to do with using the lactose that's present, okay? So we have induced the Lac Operon so that it will transcribe and translate to produce these products. Okay, so a Lac Operon is an inducible operon. If there is certain thing present there, in this case lactose, then it will turn on those genes, right? And this is not a special thing about lactose. It's not unique. We find Lac Operons, glucose operons, sucrose operons, all sorts of operons. And each one of them would turn on the appropriate set of genes if that sugar is present, okay? Now, some bacteria prefer certain sugars, and they will prefer to turn on certain operons to use those sugars. So let's look at this graph here. Here we have a graph showing the growth of E. coli cells. And if you're watching this before the next, the second module exam, this is a little bit of review for you. This looks like a little bit of a lag phase, right? Lag phase, you put these cells into a tube, and let's say that tube has glucose and lactose in it. Well, it so happens that E. coli prefers to use glucose, okay? So here you can see that there's going to be growth of the cells, and it's using up the glucose during this time. Now let's say it gets to a point where it's used up all the glucose in that tube. Well, we see again that these cells are going to kind of revamp, right? They're going to equip themselves with other enzymes that will allow for the use of lactose. So here you can see literally a pause during the growth of these E. coli cells where they shifted to turning on the lactose operon and using that lactose. Okay, so it's a pretty cool function, this idea that we can turn on these genes at will. Okay, let's look at one more kind. So we've done our inducible operon. Let's look at a repressible operon. Okay, this is called a trip operon. This TRP here stands for tryptophan, and that happens to be an amino acid that can be incorporated into proteins. Now, tryptophan is actually a really expensive amino acid to make. It has sulfur in it, and sulfur isn't always a plentiful element, depending on where it is this organism is living. So we don't want, this particular set of genes produces tryptophan, and we don't want to frivolously make tryptophan because it's so expensive, right? These sulfurs are hard to come by. So we want to very frugally turn on the tryptophan operon to make tryptophan. We don't want it on all the time, okay? And the difference here is with Lac Operon and sugar operons like that, it's if the sugar is present for the use of the sugar as a nutrient source. Here, it's for the production of a product that will be used in proteins, okay? So here we see, again, a basic structure of an operon, right? Promoter, operator, bunch of genes that are linked in their production of tryptophan in this case, and we have a repressor protein. Our repressor protein will get transcribed, as always, right? So here we have our mRNA. We'll have translation of that repressor with that mRNA to make the protein. Now, when the protein was produced in the Lac Operon, it was an active repressor protein to start with. Here, the native shape of the repressor protein is inactive. It is not capable of binding on the operator when it's made, right? So in this particular case, it would not bind to the operator. The RNA polymerase would bind at the promoter to get started with its transcription process, and there's nothing to stop it. It's going to go right on through that operator and produce each of these proteins. In this case, those proteins will aid in the production of tryptophan. So we have tryptophan synthesis. Okay? All right, so the native situation is that we'll make the tryptophan. With the Lac Operon, the native situation was that we would not make the products, right? Here, the native situation is that we will make the products. All right, let's look at the trip operon, but now let's say that there's plenty of tryptophan in the tube, okay? We'd have the mRNA for the repressor produced. We'd have an inactive repressor protein to get started. But now what happens is tryptophan, which is this molecule right here, binds to the repressor, which is currently inactive, and it's going to make it an active repressor protein. Now, when there's a lot of tryptophan around, this active repressor protein is going to block the reading of these genes. Now, this makes perfect sense, right? If there's a lot of tryptophan around, why should I make any, right? So this active repressor protein being triggered by the tryptophan is a perfect situation, right? Does exactly what we want it to do. All right, so we'll call it quits for now. And next time we'll zoom on into, let's see, what are we getting into? Looks like we're going to probably get into some mutations, okay, and what that does.